Thanks everyone for joining us from all parts of the US and elsewhere. Before I get started, I'm just going to mention that the work I'm going to talk about is a part of a long-term study, and we've been funded by a lot of different sources over the years. List a few of them here. It's really a team effort. A lot of folks that are working on photo identification catalogs or data analyses in the office, really important team of folks in the field. And a lot of the work that we've been doing is collaborative in nature, and collaborations with folks from Pacific Island Fisheries Science Center, Southwest Fisheries Science Center, Florida International University, Pacific Whale Foundation, and other groups. So it's really a team effort that allows us to accomplish as much as we have. I also need to mention that citizen science or community science has played a really important role in understanding the different populations of dolphins and whales around the Hawaiian archipelago. This is just a word cloud of folks that contributed photos up until 2019, and you can see both a large number and if you recognize some of the names, it's really a very diverse group. Folks who work in the tour industry, people who are professional photographers or naturalists, and fishermen, and just a wide variety of people that are contributing. So just to set the stage, the Hawaiian archipelago is the most isolated archipelago in the world. This shows the blue line represents the 200 nautical mile limit surrounding the archipelago. It's located almost in the middle of the Pacific Ocean. Now, if we look at it from an oceanographic perspective, this map shows chlorophyll A, which is an indicator of primary productivity. And the Hawaiian archipelago is located pretty much right in the middle of this large blue area, and that represents an area of very, very low productivity. Now, if we zoom in on the archipelago itself, and you look particularly around the northwestern Hawaiian Islands, up here you can see these hotter, warmer colors, reds and yellows, that represent increased productivity around the islands. And around the main Hawaiian Islands as well, you can see that increased productivity. So basically that increased productivity represents that the islands themselves are like an oasis in the middle of a vast biological desert. And this oasis is created from a number of different factors. Rainfall is important. This is a rainfall map that shows rainfall among the islands. And you can see that on the eastern and northeastern parts of the island, there's increased rainfall. That rainfall washes a lot of nutrients into the water. The winds in Hawaii predominantly come from the west, from the east and northeast, blowing to the west. And the winds are driving currents, they're driving upwelling, and they're also driving eddies that form off the leeward sides of the islands. So you can see off the north end of Hawaii Island, there's a cyclonic eddy and causing upwelling. You can see anticyclonic eddies that cause downwelling. And these eddies basically increase local productivity. They're ephemeral, they propagate away from the islands, but they're also either bringing cold, nutrient-rich water up to the surface, or they're concentrating prey in the middle of these downwelling eddies. Now, I first moved to Hawaii in December of 1998, and when I arrived there, with the exception of Dan McSweeney in Kona, all the researchers that were studying whales and dolphins were either studying humpback whales or spinner dolphins. I didn't want to overlap with other researchers. I didn't want to compete with them. So instead of focusing on those two commonly studied species, right away my interest and my time was spent working with other species. So in the upper right, false killer whales, species that I've been interested in for a long time, since having a stranded animal in British Columbia in the late 1980s that I had the opportunity to work with others and conduct a necropsy on. Bottlenose dolphins in the lower right, pantropical spotted dolphins in the lower left. And when we were able to get offshore to the west of Lanai, we'd also encounter pilot whales. Now, this initial relatively short foray to the islands in 98, 99, basically evolved into a long-term study. It's been ongoing every year since 2000. Over the last 22 years, we've had 1,250 days in the field, which is about 57 days a year. Typically, this involves going out to the islands 2 to 3 times a year for 2 to 3 weeks at a time, and being able to spread the effort out, not just off of Maui, where I started, or Hawaii Island, where we've worked every year for the last 20 years in a row, but with some effort off of other islands as well. Off of Oahu, we've worked 6 different years. Off of Kauai and Niihau, 13 different years. Maui Nui, 9 different years. And I'll also mention that during COVID, we've basically adopted a new strategy that's was basically a response to the travel shutdowns and the uncertainty associated with COVID. One of our main team members, Colin Cornforth, is based in Kona, and when the pandemic hit, he basically started going out and doing a rapid response type of field effort. So when folks would report false killer whales or other high priority species, he would basically head out and work with those animals. So that's been a new change just in the last couple of years. Now, during our regular field efforts, we are trying to, we're not doing blind transect surveys like many other researchers. We're trying to maximize encounters with different species. And we do that primarily by trying to remain in areas with good working conditions. With the trade winds, it's very easy to, if you had sort of standard line transect lines to run, it'd be you'd spend a lot of your time in areas that were just way too rough to really accomplish anything. So we try to spend our time in areas with Beaufort 2 or less, so try to avoid whitecaps. That said, we're also trying to maximize the spatial extent of where we survey. So this map shows 3 different days of survey lines. One day where we went north, one day where we went south, and one day when we went offshore. And we do that as much as we can, as well as diversifying our effort as much as we can in terms of depth. Within a particular field project that may last 2 or 3 weeks, we try to minimize the overlap in the survey track lines and also try to reduce any other biases. So we're forced by the weather to go south multiple days in a row. We may head south the first day in 800 meters depth and come back north the next day in 2,000 meters depth. And then the day after that, we may go south in 3,000 meters and come back up north in 1,500 meters. So we're trying to minimize any sort of biases in our work. Now I'll show you a little video here that shows a couple of our main techniques. So there's 3 different photographers on the boat here. This person in the bow is Daniel Webster. He's holding a air rifle, and this is a false killer whale off of Kauai, and they're trying to get a tag deployed on the false killer whale. Also note that on the lens with the GoPro, you'll see 2 lasers that are green dot lasers for measuring animals. That shows a deployment of a tag on this false killer whale. So I showed in the last map that most of our survey effort was on the west sides of the islands, the leeward sides of the island. But this tagging work, and I'll show you one of the tags here to give an idea of the size. This tagging work basically is a way for us to monitor where the animals go outside of our study area and for much longer periods of time than we would otherwise be able to study them. The last few years, we've also started incorporating land-based observations into the work, and these have been done in conjunction with boat-based efforts. So we have a boat on the water as well as a land-based team, and the land-based team is using tripod-mounted binoculars and tripod-mounted spotting scope to both detect groups and vector the research boat into them. These photos are from a project off Lanai in December 2020. And if you look on the right-hand side, that land-based site is right up at the very top of that cliff. 206-meter high cliff, and our boat in this particular drone image is down at the bottom of the cliff. So it gives you an idea of the spatial scale. And these land-based or combined land and boat-based efforts have really allowed us to increase our encounter rates, as well as basically allow the flexibility of the research vessel to go and search elsewhere when the land-based team is not spotting the species of interest relatively close to shore. So over the years, again, it's a long-term study. We have a lot of different type of information. We have sighting data, over 3,500 sightings from 21 different species, and that comes from over 140,000 kilometers of effort. We have photo identification catalogs of 15 different species, 13 species of odontocetes and 2 species of baleen whales. Some of these catalogs are quite small, 24 individuals. I think that's our sei whale catalog, up to almost 2,500 individuals, our pilot whale catalog. We've also been collecting biopsy samples pretty consistently over the years. We have samples from 15 different species, over 1,500 samples, plus prey samples, slough skin samples, breath samples, and fecal samples, and those samples are really used for a wide variety of studies. Since 2007, we've been using those limpet satellite tags that I showed the video, and we have data from 12 different species over 350 deployments. These range from just a couple days to over a year, but the average is about 3 years, or 3 weeks, sorry. We also have a subset of those tags are recording dive behavior, gives us dive depths and dive durations, and we have dive data from 7 different species, over 100 deployments. Back in the early years, and you can see a picture of one here on this juvenile Blainville's beaked whale, we were using suction cup attached time depth recorder tags to look at diving behavior. We have data from 50 deployments on 6 different species. In recent years, we've started deploying drones for a variety of purposes. Some of the drones have been carrying a laser altimeter on board, which can be used to measure individuals, and others are just to record behavior or even to collect breath samples. And we have drone footage from 13 different species. And lastly, we're recording information on other vessels we see out in the water, seabirds, flying fish, bait schools, game fish, all of these things to try to give us a better idea of what's going on in the environment of the different species of whales and dolphins that we're seeing. Now I'll show you an example of how we can use the sighting and effort data to just make some basic conclusions regarding spatial use. This shows 3 different species. The yellow lines represent all of our survey track lines off of Kona. On the left-hand side, these are sightings of rough-toothed dolphins, which are generally deeper than about 1,000 meters depth and basically extend all the way offshore. In the middle, these are short-fin pilot whale sightings, which are actually the most frequently encountered species in our work. They are also limited in terms of the inner, shallower component of it, but unlike the rough-toothed dolphins, they're very, very concentrated in slope waters between about 1,000 and 2,500 meters depth. And then you compare on the right-hand map that shows bottlenose dolphins, which are found very, very close to shore, primarily in less than 1,000 meters. Now, even with very non-systematic survey effort, when we're doing these types of comparisons among species, we can pretty we can come up with detailed comparisons or detailed understanding of the habitat use of these species. I will point out for the bottlenose dolphins that there is one point very far offshore in over 3,000 meters of water. And in that case, that particular group of animals represents an open ocean population of bottlenose dolphins. They're much larger animals, much more scarred up, and from our photo identification work, there were no matches to our catalog. So it represents one of the rare pass-bys of open ocean bottlenose dolphins past the island. Now, even with some very infrequently encountered species, we've been able to very slowly gather enough information to make some, I think, important conclusions about their fidelity. This is an example from a paper that came out last year on dwarf sperm whales. We've always had this approach where how much time we would spend with a species would really be in inverse proportion to how often we would encounter it. So a species like dwarf sperm whales that we would encounter very rarely, we would spend as much time with as possible. This is one particular very distinctive individual in our catalog. We've actually documented this individual over a 15-year span. And the map on the right shows the locations of sightings of that individual. And the locations are all fairly clustered compared to our distribution of sightings of dwarf sperm whales in general, which really reflect that individuals in this resident population have a very limited home range, very small home range. Now, other types of information we're able to get from our photo identification catalog with dwarf sperm whales. One thing is that the species seems to be regularly attacked by large sharks. These individuals on the left-hand side, the three photos here are all of the same individual, which had survived a massive shark bite wound, although this individual has not been resighted off the island, so it may have survived just for a short period. But these other three individuals also have shark bite wounds, this one on the back, one on the dorsifin, and another one on the dorsifin. That suggests that dwarf sperm whales are targeted by sharks fairly regularly. We're also, even for a species that is extremely difficult to get close to, we're able to document evidence of interactions with humans in some ways. This individual here on the right-hand side, it's either a line injury or a propeller strike wound. Other individuals have evidence of entanglements. So species that you wouldn't think of necessarily interacting with humans much is still impacted by our activities. We also document evidence of marine debris entanglement or fishing gear interactions through our photo identification catalogs. And importantly, we're able to use the catalogs to assess the outcomes of those interactions. So this short-fin pilot whale, for example, with a line trailing from the mouth, we've documented this individual multiple times since, as recently as last year. Or the year before, so we know that whatever happened with this entanglement did not cause any sort of long-term effects. On the other hand, this Blainville's beaked whale that we encountered last year, entangled in a piece of marine debris, there have been no sightings of this individual since, and we suspect that it's died. This is one of the reasons why we really encourage citizen science contributions. Even one encounter of a species like Blainville's beaked whale can play a really important role in helping understand things like how animals are surviving or not surviving from these types of human interactions. Now, this is that same Blainville's beaked whale with the cargo net wrapped around the head. You can see the bottom photo taken from a GoPro video when we first encountered it. We actually had several prior encounters with that individual contributed by citizen scientists in the couple months prior, and we're able to use those photos to try to get a better idea of how long was this animal entangled when we encountered it. It wasn't able to open its mouth based on the netting around the head, and we're able to use these images to assess body condition and changes in body condition in the not quite 2 months between these two encounters. And it suggests in this case that the animal's actually lost a fair amount of body mass, and that entanglement, while it looked fresh to us, may have been on the animal for several weeks. We're also trying to understand other types of human interactions. There's not a lot of aquaculture in Hawaiian waters, but there is one fish farm off the Kona coast, and we've been documenting, along with a lot of citizen scientists, interactions with a number of different species with that farm. This photo actually shows a false killer whale passing by, but in particular, the interactions have primarily involved bottlenose dolphins, and they first began to appear at the site shortly after the farm went in. There were some early reports of farm staff that were feeding bottlenose dolphins. And through our photo identification work, we've been able to document that about a quarter to a third of the entire population of resident bottlenose dolphins is actually interacting with that fish farm, and the numbers seem to be increasing over time. And this is particularly important because this farm is located fairly close to a traditional spinner dolphin resting site, and there have been a number of cases of those farm-associated dolphins harassing or potentially even killing spinner dolphins, and may have resulted in an abandonment of that resting area. So I'm going to give you a few examples of ways we're using photo identification data to better understand the populations in Hawaii. This example is with melon-headed whales and was the work of Jessica Ashatino for her master's degree. Now on the right-hand side shows a social network. Each of these points represents an individual, and the lines, when there's so many of them here, basically show associations among individuals. So for example, this small cluster, these three small clusters were all groups that were just encountered on a single location and were not seen again. This largest cluster in the bottom represents probably dozens and dozens of encounters, and this cluster up here represents more than a dozen encounters as well. Now, what we would have expected with these groups that are isolated, these three isolated clusters, is not surprising. Relatively few individuals were photo identified in any of those clusters, but the fact that there's no links between the two main clusters turned out to be a very surprising finding in the study. When you look at the location of the encounters from this large cluster, individuals within that group were documented moving between Kauai, Oahu, and Hawaii Island, whereas this smaller cluster, but still involving more than a dozen encounters, all of those individuals were off of Hawaii Island. When you look at the details of the sightings in depth, all of those Hawaii Island only encounters were in very, very shallow water, average of 381 meters, whereas the larger cluster was in much, much deeper water, those encounters. And this work, this photo identification work, actually led National Marine Fisheries Service to recognize two different stocks or two different populations, one called the Kohala resident stock, which probably has the smallest home range of any population of whale or dolphin in Hawaiian waters, and the Hawaiian Islands stock. And we now know as well from other types of evidence, including satellite tagging and genetic work, that these two populations are genetically differentiated and they're utilizing very, very different areas. Another paper that we had come out more recently, this was a paper published in Endangered Species Research last year, uses our photo identification catalog of bottlenose dolphins to look at abundance and trends among the islands. This is a larger collaborative effort with folks from Pacific Whale Foundation and Wild Dolphin Foundation. And what we found is that there's some evidence of a decline in abundance off of Kauai and Niihau. There's some evidence of a decline off of Oahu, but importantly, there's strong evidence of a significant decline off Maui Nui. We know from prior studies that these populations are all demographically isolated. There's very little interchange among them. And this Maui Nui population decline was a real surprise for all of us, and really leads to a lot of new questions as to why is that population declining in comparison to the others, and do they have different toxin loads? Do they feed on different things? They have a reduced body condition, so it leads to a lot of new questions. So now I'm going to switch to false killer whales and talk about how we can combine the social network information, the genetics, and the tag data from our satellite tags to inform stock structure of these populations. Now, unlike the last example with melon-headed whales showing a social network, here we see a lot of these isolated clusters, sometimes with just a few links. You can tell that some of them just represent a single encounter just based on the number of individuals and the symmetry of those associations, whereas others represent multiple encounters. Now, this large cluster of at the bottom, we've had a number of individuals in this group tagged, and this social cluster basically represents what we now call the main Hawaiian Islands population. This map shows tag data from 40 different groups over about a 12-year period. And you can see that they're all restricted to the main Hawaiian Islands out to about 100 kilometers offshore, and a number of areas of concentration among them. Now, if you look at another one of these social clusters, here is Oahu in the bottom right of this map, Kauai and Niihau here. These individuals have either been tagged off Kauai and Niihau or up in the northwestern Hawaiian Islands. And unlike the main Hawaiian Island population, this appears to be an insular northwestern Hawaiian Island population that ranges as far west as Gardner Pinnacles up here. Now, we've also tagged individuals in 4 of these isolated clusters. And unlike the main Hawaiian Islands or northwestern Hawaiian Island individuals, these have ranged over very, very large areas. So this shows the entire archipelago, and again, the 200 nautical mile of economic zone outlined here. And you can see that these false killer whales are truly international. They're ranging over 1,500 kilometers east of the main Hawaiian Islands and into offshore waters, as well as throughout the archipelago. And this is what we now know as a pelagic population. Now, if you look at the genetics, we know, and this is a median joining network of mitochondrial haplotypes. Mitochondrial haplotypes are inherited from mother to offspring. So if it's a mother and an offspring, they have the same mitochondrial haplotype. And these are color-coded by where they were collected. And what you can see is that the main Hawaiian Island population have very, very closely related mitochondrial haplotypes, and almost all of them are just one of these 2 haplotypes. Now, the northwestern Hawaiian Islands animals lump in with the main Hawaiian Islands, and that basically shows that these insular false killer whales in the islands came from the same colonization event that occurred probably many thousands of years ago. Now, the animals sampled offshore and elsewhere in the Pacific and the Atlantic have a really wide diversity of haplotypes that are quite distinct from the main Hawaiian Island and northwestern Hawaiian Island animals. So what this shows is that these animals are socially isolated from the social networks. They have different ranges from the tag data, and these ranges and social isolation has been persistent for long enough for the populations to diverge genetically as well. Now, the main Hawaiian Island insular population is probably our highest priority species and highest priority population of all the species that we study in Hawaii. It was estimated to number almost 500 individuals back in the 1980s, but there were declining sighting rates in aerial surveys and boat-based surveys really from the 1990s through the 2000s. And we first estimated abundance of individuals from this population in 2005 using our first 5 years of photo identification data, and came up with an estimate of just 123 individuals. Now, the NRDC petitioned NIMS based on a whole variety of information, the evidence of decline and the small abundance, to list this population as endangered. National Marine Fisheries Service undertook a status review in 2010 and recognized it as a distinct population segment to the Endangered Species Act, and then listed it as endangered in 2012. And a more recent abundance estimate from 2015, it's actually 167 individuals, but we have a good reason to believe it hadn't increased from the 123 we estimated a decade earlier up to 167. Instead, we've just gotten a better grasp of how many different social groups there are in the population. We're currently working with Pacific Island Fisheries Science Center and Pacific Whale Foundation to come up with a new abundance estimate. Now, like killer whales along the west coast of North America, we know these individuals travel in long-term groups, stable groups of related individuals. We know that from genetic work as well as the photo identification work. We haven't called them pods, but instead call them clusters, simply because of the clustering that occurs. This shows with some restrictions on the data, and we have evidence basically of 5 more than others, and some of which are fairly isolated and rarely interact with other social clusters. Now, one of the reasons why the population declined so dramatically between the 1980s and the 2000s probably relates to persistent organic pollutants. Like killer whales, false killer whales are upper trophic level predators. They feed on a lot of predatory fish. And we published this paper in 2020 that basically shows that in terms of PCB levels, basically every adult male in the population has PCB levels that are high enough to potentially cause immunosuppression, and over half the adult females in the population have levels that are high enough to cause immunosuppression. And that's not surprising given what they feed on. I mentioned they're upper trophic level predators. We see them feeding on large pelagic game fish like this ono or wahoo, mahi-mahi. This is a broad-billed swordfish in the bottom left, opa in the bottom right. And if you look at the list of known prey, with the exception of a couple species of squid that have been documented only from stranded animals, and one filefish, basically everything that false killer whales in Hawaii are feeding on are species of fish that are also targeted by humans, including a lot of pelagic game fish like yellowfin tuna, big-eyed tuna, skipjack tuna, moonfish, mahi-mahi, and so on, but also reef-associated game fish like this alua or giant trevally. Now, we've known that fishery interactions have been ongoing at least since the 1960s, and they primarily involve false killer whales taking fish off of lines. The first conclusive evidence of this comes from 1963, and it was mentioned in Karen Pryor's book, Lads Before the Wind, that the catcher for Sea Life Park on Oahu went over to Kona, went out with a longline fisherman, and observed false killer whales taking catch off their lines. In the 1970s, this type of behavior of depredation, taking fish from fishermen, was considered to be very common. And it's likely these interactions that were responsible for the majority of the decline in population. There's a lot of different types of fishing gears used in Hawaii, a lot of different type of fishing activities. And the largest fishery in terms of the total amount of catch comes from the Hawaii-based longline fishery. Now, through the late 1980s, this longline fishery increased dramatically. This graph shows the number of longline vessels in 1987 was less than 40, and by 1990 was 140. And the number of longline vessels has remained relatively constant since then. Now, back in the early days, this longline fishing was occurring very close to shore, as well as offshore. In 1991, there was an exclusion zone put in off the northwestern Hawaiian Islands. And in 1992, there was another exclusion zone put in around the main Hawaiian Islands. And this map that's showing on the right shows these exclusion zones around the main Hawaiian Islands. for a long time, the exclusion zone around the main Hawaiian Islands would shrink towards the islands for 4 months of the year. Now, this main Hawaiian Islands exclusion zone for longline fishing was put in really to minimize conflicts between the nearshore fisheries and the larger longline fishing vessels. And that said, even after the exclusion zone went into effect, there's a lot of commercial fishing effort around the main Hawaiian Islands. Now, bycatch primarily in the offshore longline fishery, the deep set fishery for tuna. There's observer program in that fishery with about 20% observer coverage. And it was known for a long time that false killer whales were the most frequently recorded species of whale or dolphin killed in that fishery. And that level of bycatch led to the formation of a take reduction team in 2010 and a take reduction plan that went into effect in 2013. Now that take reduction plan basically excluded longline fishing year-round from around the main Hawaiian Islands. It's shown here. It got rid of that 4 months of the year where fishing would occur closer to the islands. And the take reduction plan also included basically mandating what were considered weak circle hooks. This shows you an example of one of the main hooks that was used, that is currently used in the gear. The idea is that the maximum wire diameter of the hook would be 4.5 millimeters, which would make the hook relatively weak. In theory, a false killer whale could get hooked on one of these, and as long as the terminal gear, the branch line, could straighten the hook and get off the gear. And there were also gear handling recommendations that went into effect to try to encourage fishermen to put pressure on the gear to increase the chances of a false killer whale getting off the hook. We now know that take reduction plan doesn't work at all. It's really been completely ineffective. The first 5 years after it went into effect, the interaction rates remained the same, and there was no significant decrease in serious injury or mortality. And in fact, 2021 bycatch rates on record. We know that the hooks aren't weak enough, the branch lines aren't strong enough, and that more training is needed because the fishermen are often not doing what they need to do in order to try to make the weak hook, strong branch line gear fix work. But importantly, the whole plan is fatally flawed because there's only observers on 20% of the boats. And what happens on the other 80% is completely unknown, but the chances of fishermen doing what they should be doing in terms of trying to put tension on the lines to straighten the hook when there's no observer on board is, I think, extremely low. Now, this map shows the bycatch records in this longline fishery from 2020. These include animals that were, a couple animals that were known to have died, a lot of animals that were considered seriously injured, and a couple that were released with no hooks or gear attached. But you can see that a number of them are in this area just south of the main Hawaiian Islands, and a lot of them are out in pelagic waters. Now, most of this bycatch is happening with the pelagic population and not with the insular false killer whale population, the one that we know is relatively small, we know is restricted to the main Hawaiian Islands. Programs and in the nearshore fisheries, and the big question is identify catch, because you've got someone, independent observer on board the boat to document how often it occurs. What are the other ways of doing so? We first published a paper trying to use dorsal fin disfigurements, indicator of fisheries interactions, back in 2005. This is actually a pygmy sperm whale, but this is a still taken from a GoPro footage of a hooked one on a long line. The animal's hooked in the mouth. You can see the trailing line here. And on the leading edge of the dorsal fin, you can see an injury. And in the video, you can actually see that this is a fresh bleeding injury. And so when an animal gets hooked on the line, it often struggles against the line, and injuries result, including in areas like the dorsal fin, where we can see that and get photographs of it. We published another paper in 2014 that quantified these types of injuries, and this is a pretty extreme example where the cut through the leading edge base of the dorsal fin goes almost halfway through the fin, and found that individuals from this main Hawaiian Island population have significantly higher levels of these dorsal fin injuries compared to individuals from the pelagic population, where we know there's bycatch, or individuals from the northwestern Hawaiian Island population. And about 9% of individuals have these types of line injuries. Now, the dorsal fin is just an indirect way of looking at injuries, and the mouth line, if the animals get hooked in the mouth, is much more likely to show evidence of hooking. And about a quarter of the individuals in this endangered main Hawaiian Island population have evidence of mouth line hooking. And this is an example where you can actually see it's missing a lot of tissue, and it has some broken teeth. So recognition that solving depredation and dealing with the bycatch issues is going to require buy-in from commercial fishermen and recreational fishermen. And there have been a lot of discussions in Hawaii about the main Hawaiian insular population of false killer whales, development of a recovery plan. But the people that have been, the fishermen that have been coming to these meetings, have basically largely been unwilling to acknowledge that there is a bycatch issue. And so we wanted to get at why that is. And the problem, I think, is that often the wrong fishermen were coming to the meetings or were being asked. And this map shows some information on fishery catch data from Hawaii. Almost a quarter of the entire catch comes from this relatively small area off Kona. When you look at our satellite tag data from false killer whales, and this basically is a way of looking at the satellite tag data in the same way as we look at the fishery data, less than 1% of their time is in that area where there's high fishery effort. So we published another paper last year that basically tries to identify which fishermen are most likely to be facing depredation and facing potential bycatch. This is a paper that came out in Biological Conservation. This map basically takes into account both the fishing effort and the false killer whale satellite tag data, and works to integrate those to come up with an assessment of from a fisherman's perspective, what's the likelihood of having false killer whales in your area when you catch a fish, which should be related to the likelihood of depredation, which should be related to the likelihood of bycatch. And it's all scaled relative to Kona. The majority of fishing efforts off Kona, we wanted to assess whether those fishermen from Kona are actually telling the truth when they say that bycatch is rare. And in fact, they are. Fishermen off Kona have a very low probability of having false killer whales around, and a low likelihood of depredation, whereas fishermen off other areas, in particular off of Kohala and off the north side of Molokai and east of Oahu, have rates of interactions, fishery overlap index interactions, that are sometimes several thousand times higher than what they are off Kona. So what does this mean? It means that if you're going to talk to fishermen about possible solutions, both to depredation and to bycatch, you need to focus on the fishermen that actually have the greatest potential for interactions, those fishing off Oahu and north of Molokai and north of Maui and north end of Hawaii Island. And there's been a lot of talk about the potential for electronic monitoring or observer program. And really, if that kind of thing is going to happen, it has to be in those areas where there's high fishery overlap indices, as I mentioned. So I'm just going to wrap up with a couple more slides and talk about some of our current work or planned work, particularly with false killer whales. We're working with researchers from Southwest Fisheries Science Center and Pacific Whale Foundation to look at the age structure of the population through epigenetic aging and drone photogrammetry. We're working in diving behavior of false killer whales using satellite tags, study on trophic ecology to try to get a better idea of how or whether diet might vary among the social clusters and how it varies among species, with Jeremy Kiska from Florida International University, looking at stress and reproductive hormones, and a few other things. We have a lot of tag data, and we're looking at how movement patterns vary in relation to oceanographic factors, as well as among social clusters. This example I'm showing you now is actually from a pelagic false killer whale. And we've also been trying to collect breath samples. I'll show you another video here. This is a drone-based breath sample collection. It's a mother and calf false killer whale. You can see it's carrying a fish in the mouth. And in this particular case, we mounted the Petri dish to the drone and are able to fly it through the exhaled breath to try to collect samples that could be used to look for pathogens, and then relate that to the photo identification data related to body condition from the drone photogrammetry, related to the prior history of pollutant loads or fishery interactions. This shows an example of the drone photogrammetry, and can again relate that to a whole variety of factors that could influence body condition. Now, I mentioned that you know, the greatest threat to this population is probably fishery interactions, and so we've also had a lot of effort to try to communicate with and work with local fishermen, and both explaining what we're doing and why we're doing it, and also trying to engage them in fishery science and citizen science. And so we actually have a program which is providing cameras to fishermen on loan if they're interested in taking photos and contributing those photos to our catalogs, and giving them information back on what they're seeing and what we're learning from it. So it's just part of our larger outreach efforts. So I think that's the last of my slides, and would be happy to answer questions.
Thank you so much for that, Robin. What a great presentation. So yeah, folks, if you have any questions at all, feel free to throw it into the chat function. There's already a question already. So once again, if you have any questions, just open up that chat function and then throw it in there, and then I'll kind of moderate the questions. So Bonnie Gertz says, hi, Robin. Do you see any evidence of orca predation on these species? I know you don't see orcas there often. Thank you.
Yeah, I mean, we've only seen killer whales in Hawaii 4 or 5 times in 22 years, and they, when they come through the islands, they are tend to stay in offshore waters. They tend to be generalist feeders rather than any sort of specialists. We've seen them interacting with marine mammals, but they've also been documented feeding on sharks, and from stranded animals, they've been documented feeding on cephalopods. We've never seen them interacting with false killer whales, which isn't surprising given how rarely we've seen them there, but we have seen them chasing melon-headed whales and rough-toothed dolphins before. And I should also say, if anyone has a question, you can just feel free to put your camera on and raise your hand as well. Otherwise, you can take a look at the comments. But there's a question about minke whales in Hawaii. We have never encountered minke whales in Hawaii, although there are a handful of recent sightings, primarily coming from one single fisherman that happens to fish out on Cross Seamount west of Hawaii Island. And he's been contributing photos of minke whales. He had one on Instagram last week of some two minke whales that were potentially feeding underneath this research vessel, but they're definitely further offshore and appear to be even more cryptic than they are when they're in nearshore waters. So I see some other questions. And I apologize if I miss your questions, because the questions may be appearing faster than I can quickly see them on the chat. And please feel free to contact me by email. Uko's question, good to see you, Uko. Do fishermen deal with extensive fishing gear damage due to depredation? And it's a good question. You know, they're basically losing, because the gear, well, let me say almost all the fisheries around Hawaii are hook and line fisheries. They're the only net fisheries are lay nets very near shore that are usually just used to catch bait fish. And so what's happening in these interactions is that they're losing hooks and they're losing lines. They're not they're not getting major damage to large expensive gear. I mean, I think that in that case, they probably lose a lot more hooks and gear to sharks or even, billfish that break free. All right, let's see what else is on the list. Please feel free to turn on your camera and ask verbally as well if you'd like. Here's a question from Jonas. Have you talked to any fishermen that fish in areas with high animal activity? And it's a good question. Fishermen, when we talk to them, are usually pretty candid on a one-to-one basis about the types of interactions they've had. I've had a number of fishermen tell me that they've lost fish to false killer whales. I've had a number of fishermen tell me they've had bycatch of a number of different species, spotted dolphins, but also a lot of unidentified species that they just can't tell what they are. Some of the fishing occurs at night, and a lot of fishermen, there's a lot of diversity of whales and dolphins in Hawaiian waters, so a lot of fishermen are just not necessarily up on how to discriminate between a false killer whale and a pygmy killer whale or a melon-headed whale or a pilot whale. So Lori has her hand up. Lori, do you want to ask a question? All right, I can't hear Lori, but let's see. There's a question about breath sampling in Petri dish. Yeah, so people have been doing breath sampling with a number of different species of whales and dolphins for several different types of analyses to collect DNA from the whales themselves, to collect stress and reproductive hormones, which are detectable in breath samples. And in our case, we were looking at the respiratory microbiome, collaborating with researchers at the Northwest Fisheries Science Center in Seattle. And what we were doing was trying to assess, so there's a couple ways you can get breath samples. If you can get close to an animal, you can put a Petri dish with some sort of sampling medium on it and hold it out on a long pole for individuals that you can approach that are bow riding or otherwise approachable. And what we were doing is using a drone basically to try to collect breath samples from individuals that were otherwise not easy to collect breath samples from. So there's a lot of species that are really hard to get close to, and certainly there's some individuals even within a species that may be interested in bow riding that won't approach boats or are hard to get close to. So we would mount a Petri dish onto the drone and fly it through the exhaled breath, and then bring it back to the boat and from the petri dish, swab it for basically droplets from the exhaled breath, and preserve those and send them off. And someone just asked whether I play a lot of ping pong. Yes, quite a bit. Have a ping pong table here in the office, and my neighbor who's in our COVID bubble and I play quite a lot of ping pong, 1,500 games last year. All right, let's get back to the whale and dolphin questions. There's a question regarding body condition for bottlenose dolphins that are at the Kona fish pens. Are they finding that they're becoming fat from the farm fish? So it's a good question. We haven't actually tried to quantify body condition from the farm associated dolphins, although I think that basically the farm itself is a really easy prey source for the animals, so it wouldn't surprise me if they had improved body condition as a result. All right, let me look at what other questions are here. Yeah, there a question regarding assistance of entangled whales. National Marine Fisheries Service and the National Marine Sanctuary, the Hawaiian Island Humpback Whale National Marine Sanctuary. Ed Lyman runs an disentanglement network in Hawaii through the sanctuary, and he's definitely the person to talk to. They do a lot of responses for humpback whales, including one last week. You know, in our case, when we found the entangled Blainville's beaked whale last year, we almost immediately got on the phone to the entanglement folks and talked with Ed Lyman and talked with folks from NOAA Fisheries on coming up with a plan to try to disentangle that animal if we were able to encounter again. And in that particular case, we spent a lot of time over the week afterwards trying to relocate it with no success. But there is a very active disentanglement network in Hawaii. Thanks for Uko for putting the comment in the chat. I did publish a book through University of Hawaii Press in 2016, which has Uko's amazing illustrations and a lot of photos from a lot of people on different species of whales and dolphins in Hawaiian waters. It's available through online retailers, etc.
So I think there was one question that I wanted you to answer, and then it's kind of with my question as well. What are you looking for when it comes down to internships and volunteers? I know that there are a lot of people that are in the group that are young who want to get into the field of marine biology or working with cetaceans. So what recommendation or tip and trick that you would give them right now?
That's a good question. I mean, I think you have to recognize that there's just a tremendous diversity of ways to get involved in whale and dolphin research. And I actually think the one thing I recommend for young people is to learn R, for programming in R, because it's a limited skill in marine mammal science and in science in general, is the ability to analyze data sets in an efficient way. You know, and I should say I don't program in R, but I collaborate with people who are really good, much more quantitative than I am. You know, getting a diversity of experience. I mean, I started off volunteering in 1985 for Thomas Learholm, who was doing some work on killer whales with Mike Bigg from the Pacific Biological Station, and then went out with Thomas for a few days and then started volunteering for Mike Bigg. And back then the skill sets that were needed were driving boats and taking photos. And not that those skill sets aren't still valuable, but there's just a tremendous diversity of ways that folks can get involved in research, and even if marine mammals are what motivates people, people should keep in mind that diversity in skills can be gained with any group of organisms, whether it's birds or fish or reptiles or amphibians, and in fact these data sets that just involve being able to manipulate data and map data and understand data. So I would say there's no one way that I would recommend other than strongly encouraging people to get proficient in R.
So I think we're gonna end it there. So if you have any more questions, feel free to reach out to Robin Baird. What's the best way to contact you, Robin?
Yeah, email is great. rwbaird@cascadiaresearch.org. I may not respond right away, and I apologize in advance for that, but that's probably the best way to get in contact. And yeah, if folks want to learn more about what we're doing in Hawaii, we try to share a lot of information on Facebook and other social media. So please join us there. So.
Yeah, okay. So thank you so much, everyone, for joining us this month for our ACS&B cetacean meeting. I'm really glad to have everyone, to see everyone again, and I hope you all have a great night.
Thank you. Take care, everyone.
Thanks, Robin. That was excellent.
Thank you.